Hello dear students, welcome to Rayat Online School Education Program of 2021-2022. Myself Professor P.B. Sargade from Sadguru Gadge Maharaj College Karad, and this is our biology's 20th lecture in this series, and from today onwards we are going to discuss about an important topic of the 12th biology syllabus, that is human health and diseases. Now this topic is very much important when we compare the syllabus and the day-to-day life of the human being. And in this topic we are going to study regarding the human health and various diseases and other points also. Now let's start this topic by continuing the content of the topic. Now we will see the content of this topic firstly, and then we will move towards the main points of this topic. Now when we consider the content of this topic has various points which are very broadly covered in our syllabus. The first point is immunity, and in that immunity there are two types: innate immunity and acquired immunity. Then we will see the structure of antibody, and the major part we are going to study is the common human diseases, and in that we will see malaria, amoebiasis, ascariasis, filariasis, typhoid, pneumonia, common cold, ringworm, dengue, cancer and AIDS. And besides these common human diseases, we are going to study adolescence, then addiction, drug abuse, and now we will start with our topic. Now before going to start the topic, we must know what is health. According to the World Health Organization, health is defined as the state of complete physical, mental and social well-being, and not merely the absence of disease or infirmity. When we study about the health of the human being, we come to various terminologies, and these terminologies, as a student we have to study these terminologies in detail, and the main terminology which comes in this topic is the immune system. Now what is immune system? Now before going to study immunity, we will study about the immune system. We will know the concept of immune system. The system which protects us from various infections or infectious agents is called as immune system. And the next term is immunity. Now the term immunity has traditionally referred to as the resistance exhibited by the host towards injury caused by pathogens and their products. When we study in detail the word immunity, it is the exempt or freedom, and this concept of immunity is believed to started by the scientist Edward Jenner in England. He developed cowpox vaccine for the protection against the attack of smallpox, which was a virus. And this term immunity originated from that study. Now we will move forward. Then next terminology we are going to study is the immunology. Immunology is a branch of science which deals with the study of immune system, immune responses to foreign substances and their role in resisting infection by pathogens. The most important characteristic of immune system is that, now listen this carefully, it distinguishes self, that means body's own cells or own body cells, and non-self, that means foreign molecules or invading cells. Now this is the very much peculiar character or important characteristic of the immune system. Now we will move forward. Now there are various terminologies again, and These terminologies are, now we are going to study the first terminology here. That terminology is antigen. Now what is antigen? Any foreign substance invading body and capable of stimulating an immune response is called as an antigen. Students, please remember this. I will explain you again. The antigen means any foreign substance which invade the body and it is capable of stimulating an immune response is called as an antigen. Now the next terminology, the antibody, and the antibody is nothing but the protective chemicals produced by the immune system in response to the antigen. Now the immunity, it is further divided into various subtypes. Now here we are going to study the types of immunity. The immunity, it is broadly classified or broadly divided into two types: innate immunity and acquired immunity. Innate immunity is starting from the birth of the individual or the human being, and the acquired immunity is nothing but it is adaptive immunity which is adapted in the life of the human being. Now the innate immunity is again divided into two subtypes: specific and non-specific. Now as the word suggests, the specific immunity, it is against the specific antigen, and non-specific immunity, it is the immunity which is in response is developed by the body. Now next is acquired immunity. We will move towards the acquired immunity or adaptive immunity. It has two types: passive immunity and active immunity. Passive immunity is further divided into natural and artificial. The natural immunity is from the mother placental circulation to the fetus. Now remember, this passive immunity develops or it goes from the mother placenta circulation to the fetus. The artificial immunity is the administration of antitoxins, antisera, and immunoglobulins. Now this was about the passive immunity. The active immunity is again further divided into natural immunity and artificial immunity. The natural immunity develops after the infection recovery after an actual disease, and the artificial immunity, it is through the immunization and vaccination. I will move towards the next point: innate or inborn immunity. The innate immunity is the resistance to infections that an individual possesses by virtue of his or her genetic makeup. Now this is most important point. This immunity or the resistance. What is immunity? Immunity is the resistance of the human being or the capability of the body for the infections that an individual possesses by virtue of his or her genetic makeup. Now this is also called as inborn immunity. Next, it is the natural inborn defense system of the body. One form of innate immunity comprises the various types of barriers which prevent entry of foreign agents into the body. Now the first barrier is the epithelial surface. This epithelial surface, it is starting from the skin, the intact skin and mucous covering of the body. Then the healthy skin possesses bactericidal activity, and this healthy skin is the first barrier for the invading germs or invading pathogens or foreign substances to the human body. And this healthy skin is a very much important factor in the epithelial surface. The sebaceous secretions and long-chain fatty acids have bactericidal activity and fungicidal properties. These sebaceous secretions help to prevent the entry of certain bacteria and fungus into the body of the human being. Now, the next most important point of the epithelial surface or the skin is that the skin also has sweat glands, and the sweat which is secreted by the sweat glands, it dries, and it contains, all of you know that the sweat contains sodium chloride NaCl. It also prevents the entry of the pathogen and other foreign substances inside the body. The next most important is the nose. The nose prevents entry of microorganisms. The nose is another entry point of the microorganisms and invading pathogens in the human body. The cough reflex is an important defense mechanism of a respiratory tract, and this cough reflex play an important role when a pathogen or the invading germ enters inside the respiratory tract. The coughing reflex throws out that infectious agent through the respiratory tract. Then next, the acidity of gastric secretions in the stomach destroys the microorganisms. All of we have studied the digestive system, and all know that the digestive system has various gastric secretions which are helpful in the process of digestion, and these secretions in the stomach destroys the microorganisms. Then the flushing action of urine eliminates bacteria from the urethra. The spermine and zinc which is present in the semen are also antibacterial. Then antimicrobial substances in blood and tissues. Now blood and tissues, they are the most important ingredients of any human body. The complement system contains more than 30 serum proteins which are circulating in the blood in an inactive state. The presence of microbial pathogens activate the complement cascade to eliminate pathogens. The interferons are a class of cytokines, that means the soluble proteins which protect from viral infection. Antibacterial substances in blood and tissues. The complement system possesses bacterial activity and plays a role in the bactericidal activity and destroys the pathogenic bacteria. The beta-lysine, it destroys anthrax. Leucines and plaquenins are there. Then lactic acid, which is found in muscles. Lactoperoxidase in milk. The next point is cellular factors in innate immunity. Now this is the most important point in our topic. The natural defense against the invasion of blood and tissues by microorganisms is carried out. The phagocytic cells. Now this terminology phagocytosis or the phagocytic cells, which were discovered by Mechnikov in 1882, are grouped as microphages and macrophages. They remove foreign particles that enter the body. The lymphocytes, they are called as natural killers or NK cells. Now this point is asked so many times to the multiple choice questions. You remember that the lymphocytes, they are also called as natural killers or NK cells, is important in non-specific defense against the viral infections and tumors. Now the next important point in this body defense mechanism is that the fever. Now the fever, what it does, it increases the body temperature following the infection as a natural defense mechanism. All of us know that when an infection occurs to our body or to the human body, the body temperature rises. Now the normal body temperature of human being is 37 degrees Celsius or 98.6 degree Fahrenheit. If this temperature increases by a certain degree, that condition we call it as fever. And this is against the response of the entry of the microorganisms or pathogens in our body. It helps to accelerate the physiological process to destroy the invading pathogens. Since the pathogens, now here one point has to remember that the pathogens, they are active at normal body temperature, it means 37 degrees centigrade or 98.6 degree Fahrenheit. And if the temperature of the body is increased to certain extent, these pathogens, they are unable to survive in the body. So the body does this mechanism of increasing the body temperature, and we call it as fever. The fever stimulates the production of interferon and helps in recovery from viral infections. The next point, acute phase proteins, or we called it as APPs. The infection on injury, it leads to a sudden increase in concentration of certain plasma proteins, which are collectively called as acute phase proteins or APPs. These include C-reactive proteins, or they are also called as CRPs, mannose binding proteins, alpha-1 acid glycoprotein, serum amyloid P, etc. APPs are believed to enhance host resistance, prevent tissue injury, and promote repair of inflammatory lesions. We will move forward. Now, these are some diagrams which are associated or which are explaining the innate immunity and the exact body defense mechanism at the initial stage. We will move forward. The mechanism of innate immunity. It is through the epithelial surface, antibacterial substance in blood and tissues, microbial antagonism, cellular factor innate immunity, inflammation, fever, acute phase proteins, and it carry over through these factors. Now the mast cells, we are going to see various types of cells in blood and others which are responsible in the immune response. The first is mast cell, in that it is against the allergy, inflammation, antigen presentation, immunomodulation. Then we will move forward. Neutrophil. Neutrophil, it is the part of WBC. It comes under the WBC, or we called it as white blood cells. It functions in cytotoxicity, phenotypic plasticity, vascular remodeling. Then macrophages, they do the function of phagocytosis, inflammation, phenotypic plasticity. Eosinophils, they are against the allergy and cytotoxicity. NK cells, they perform the function of cytotoxicity. Now this is a diagram exhibit showing the barriers to invasion. Now here it is shown that the foreign material which is entering inside the body, it is how it is tackled inside the body. It goes through various immunities: adaptive immunity, which is acquired, it is cellular cytotoxic, and humoral immunity through the antibodies. Then innate immunity, which is natural, I have explained earlier. It is through the macrophages, neutrophils, eosinophils, basophils, and monocytes, as explained in the previous diagram. And the complement system. The non-specific physical and chemical defenses or the barriers are skin, mucous membrane, stomach acid, lysosomes in tears, and expulsion through coughing, sneezing, vomiting, and through the diarrhea. These are the barriers to invasion. Now we'll move towards the defense mechanism. Now this is the most important point when we consider the human defense mechanism against the infections and again the invasions of the pathogen. It is again further divided into two parts, non-specific and specific. Non-specific, it is again divided into three parts: barriers, complement, and phagocytosis. We have discussed the phagocytosis. The barriers, as discussed earlier, through skin, mucosa, chemical through the gastric acid secretion, skin secretions, digestive enzymes. The complement through the plasmoproteins, and phagocytosis by the phagocytic activity. The specific is having two types, natural and artificial. It is antibody mediated, and cell mediated. It is through the B lymphocytes and plasma cells, T lymphocytes and macrophages. Then this is the structure of antibody we are going to study in our next lecture in detail. We'll move forward. The acquired immunity, the immunity that develops during your lifetime. It is again divided into active immunity and passive immunity. Active immunity, it is of two types, natural immunity and artificial immunity. The natural immunity, it is antibodies developed in response to an infection. This is the natural immunity of the active immunity. The artificial immunity is the antibodies which are developed in response to a vaccination. Nowadays, we are going to see various activities against the coronavirus infection, and that we oftenly heard a word, vaccination, and we are going to study the vaccination process in our next lecture. But the artificial immunity comes through the vaccination. The passive immunity, it is again also divided into natural and artificial immunity. Natural immunity, it is by the antibodies received from mother through breast milk. It is also called as colostrum. The artificial immunity, it is by the antibodies received from a medicine, from a gamma globulin injection or infusion. The immunity, as discussed earlier, it is this flow chart shows the various types of immunity. Now the acquired immunity, the resistance that an individual acquires during life is known as acquired immunity or adaptive immunity or specific immunity. The unique features of acquired or adaptive immunity. The first is specificity. Now I will explain in detail the specificity. It can produce specific antibody or T lymphocyte against a particular antigen or pathogen. The next point is diversity. It can recognize a vast variety of diverse pathogens or foreign molecules. Then discrimination between self and non-self. It differentiates between own body cells or self body cells and foreign or non-self molecules. Then the next point is memory. Now this is a very much important point, memory. The memory, when the immune system encounters a specific foreign agent for the first time, it generates an immune response and eliminates the invader. This is called first encounter. The immune system retains the memory of the first encounter. As a result, a second encounter with same pathogen brings about quicker and stronger immune response, and it comes under the memory or the unique feature of acquired adaptive immunity. I will move forward. Unique features of acquired or adaptive immunity. Specificity, we have studied. Produce specific antibody against a particular antigen. Diversity, recognizes a vast variety of diverse pathogens. Discrimination between self and non-self. Then memory. Next, types of acquired immunity. Acquired immunity has active immunity, then passive immunity. The active immunity, it is the resistance developed by individuals as a result of an antigenic stimulus. It is also known as adaptive immunity. Active immunity may be natural or artificial. Then the active immunity has 2 points: natural acquired active immunity and artificial acquired immunity, active immunity. The passive immunity, it is again subdivided into natural acquired passive immunity and artificial acquired passive immunity. We'll move forward and see all these types of acquired immunities in this tabular form. Now you are going to see it in detail. We will move towards the active immunity. The active immunity, the natural acquired active immunity, it is developed after entry of pathogens in the body, and it is a long-lasting immunity. For example, a person who has recovered from an attack of measles develops natural acquired active immunity to measles for the lifetime. And when we consider the passive immunity, the natural acquired passive immunity, it is before birth maternal antibodies are transferred from mother to fetus through placenta. The natural acquired passive immunity is short-lived. It is for a shorter span of time. After birth, antibodies are transferred from mother to infant through the colostrum. The colostrum is nothing but the first milk of mother, which is very much important, and the doctors, they advise to breastfeed the newborn babies the first milk of the mother because of this, because of the colostrum, which contains the antibodies, and it continues throughout the period of breastfeeding. The artificially acquired active immunity, it is the resistance induced by vaccines. Vaccine is introduced into the body to stimulate the formation of antibodies by the immune system. Example, polio vaccine, BCG vaccine, etc. Such immunity may be temporary or permanent. Now, this was about the artificially acquired active immunity. Then, artificially acquired passive immunity. This immunity is developed by injecting previously prepared antibodies using serum from humans or animals. Now, the serum is used which is containing the antibodies from the humans or animals. For example, the antibodies obtained from hyperimmunized horses are injected to humans against rabies pathogens, and it is short-lived. We will move towards the next point. The cells of immune system. Now, there are various cells of immune system we are going to study. In this, lymphocytes, then antigen presenting cells. Now, in lymphocytes, the lymphocytes are the main cells of the immune system. Mechanism of response of T lymphocytes to antigens. It is through the helper T cells, and this is the diagram showing the helper T cells. These helper T cells, these are the sensitized helper T cells which produce lymphokines for performing several types of functions like proliferation of other T cells, stimulation of B lymphocytes, and macrophages, etc. Then we will move forward. We are going to see the diagram of killer T cells. Now, these killer T cells, they are also called as cytotoxic T cells. Now, why they are called as cytotoxic T cells? They directly attack and destroy the invading microbes, infected body cells, and cancer cells. The killer T cells or cytotoxic T cells bind to infected cell and secrete perforins. Then perforins forms a hole in the infected cell. It also releases substances that kill the cell. Hence, the name is also called as cytotoxic T cell. Now, the next third is suppressor T cells. The diagram is in front of you. Now, the suppressor T cells, these are the cells which suppress the entire immune system against attack on the own body cells. Now, these are also important cells when we consider the mechanism of response of T lymphocytes to the antigens. And now we will move towards the next, memory T cells. The memory T cells are also important, and the diagram is in front of you. These are previously sensitized cells which retain the sensitization memory for a long time in the future. Mechanism of action of B lymphocytes to antigens. They are sensitized directly to both antigens as well as by helper T cells. The main functions: free antibodies, then agglutination of particulate matter including bacteria and viruses. The immunized mass is then engulfed by phagocytosis. Second, opsonization or coating of bacteria to facilitate their subsequent phagocytosis by macrophages. All of us know that the phagocytosis is carried out by microphages and macrophages. This action is carried out by the macrophages. Then third, neutralization of toxins released by bacteria. Example, tetanus toxin. Now, these are the diagrammatic representations of the actual process of the mechanism of the immune response by the cells. Now we will move forward. The antigen presenting B antigen presenting cells are antigen presenting cells engulf invading pathogens and process the antigens. Then the processed antigens are presented on their own surface. Then these cells are able to deliver a stimulatory signal that is necessary for activation of helper T cells. And by this way, the invading antigen, it is destroyed or it is tackled, it is neutralized in the body of the human being if it enters in the body. Thank you. For this topic, we have completed up to the mechanism of action of B lymphocytes in the body, and the point includes the entire mechanism of the body defense mechanism. We will stop here and we will see the next point in next lecture. Thank you all of you.